We're now going to discuss the cyclic equivalent of, the cyclic amides. So it's the amide equivalent of the lactone we discussed earlier. Cyclic amides are called lactams. And in fact, they're classified the same way we classify lactones. In other words, we look at the carbonyl carbon, we go to the adjacent carbon, we count alpha, beta, and so forth. Whichever carbon is connected to the nitrogen and then connected back to the carbonyl, that's the carbon that we use to designate our lactam. So for example, a four-membered ring lactam would be alpha beta, nitrogen C double bond O. That would be a beta lactam. 5 would be gamma lactam. 6 would be delta lactam, and so forth. Beta lactams are extremely interesting because they are the structural basis for the class of medicines called penicillins. I'd just briefly like to discuss with you how a penicillin molecule functions biologically and why that makes it an antibiotic. So, why do we need an antibiotic like penicillin? Well, the problem is that when we have a bacteria like this, our natural defense, in other words, our immune system, works in this way. It recognizes the bacterial cell as a foreign cell. It directs our immune system cells to attach to that bacterium and cut open the membrane of the bacterium. Because the bacterium is a single-celled organism, when you cut open the membrane of a cell, you effectively kill the cell because it releases all of the important metabolic molecules that the cell needs to survive. So if we cut open the bacteria, we kill the bacteria. As a result, there have been bacteria that have evolved a protection from our particular defense mechanism. What they do is they build around themselves what is called a cell wall. Cell wall is essentially made out of carbohydrates, made out of sugars. And in fact, it effectively is similar in terms of its composition to wood. The cell wall is built with small little openings that allow nutrients, water, air, all of that kind of stuff to flow in and out, but will block the larger defensive cell from our immune system from being able to get in and cut open that membrane. So in a sense, cell wall is like armor for the bacteria. This makes these bacteria particularly dangerous to our organism because if they get in, if they infect us, our natural defenses can't kill them. And so they will essentially keep reproducing until they overwhelm our body, absorb all its nutrients, and essentially kill us. So in the past, before antibiotics, these type of bacteria were literally deadly. In fact, they accounted for a large number of fatalities tragically from just very simple types of injuries that got infected. The penicillin molecule was one of the first modern antibiotics that was discovered, and in fact, it was discovered by scientists who took these bacteria, they grew them in petri dishes, and then they just started putting in different types of substances, extracts from plants, from molds, from all sorts of stuff, to see if any of them stopped the bacteria from growing, from reproducing. And they did, in fact, that there was find that extracts from a certain type of mold were effective at stopping the bacteria, this type of bacteria, from growing. They then took those extracts, they did the typical kind of lab work that we would do, extractions and things like that, isolated the extracts, and discovered that there was a specific molecule in them, a penicillin, which was responsible for their biological activity. Penicillin molecule looks something like this. I've abbreviated it somewhat. This group that I've abbreviated R actually has a number of different structures that build off of this, And so there are a number of different penicillin molecules. There's penicillin A and G and H and all of these kinds of things. And they differ just in what is the identity of this carbon group. But all of the penicillins have preserved this basic core structure right here. And if we look in this core structure, we can see that there is a beta-lactam. Here's a carbonyl, alpha carbon, beta carbon. Now, it's considerably more complicated than just that. We can see we also have attached to this side of the lactam a second ring, which has a sulfur in the ring, a 5-membered ring, and then we also have a carboxylic acid right here. And it turns out that this structure altogether combines to form kind of a mousetrap that enables this molecule to react with the enzyme and essentially inactivate it. Now, what enzyme does it react with? Well, the bacteria use an enzyme, which is a large protein. They use an enzyme to catalyze the reaction that builds their cell wall. It turns out that this enzyme has on its surface many nucleophilic atoms and groups that could react with a suitable electrophile. For example, a carbonyl carbon. So, the enzyme is present once the bacteria is present. We put in the penicillin. That enzyme reacts with the penicillin molecule by a typical SN-AC reaction. So the nucleophile makes a bond to the C double bond O carbonyl, pushes a pair of electrons out. We get this structure. This is a tetrahedral intermediate. Now, here's the interesting thing. Normally, we would not expect this tetrahedral intermediate to be able to push out this nitrogen. As we talked about, nitrogen negative is a very, very weak leaving group because it's such a strong base. However, this particular molecule is totally designed so that that nitrogen will become an excellent leaving group when it's needed to be. There's two things that drive this. First of all, if this tetrahedral intermediate pushes out the nitrogen, it breaks open the 4-membered ring. And what we know about 4-membered rings is that they have ring strain. So by pushing out that nitrogen and breaking open that 4-membered ring, we release an enormous amount of potential energy, which drastically reduces the amount of energy that's going to be required to push this nitrogen out. The other thing we know is that we can make nitrogens like this better leaving groups by protonating them. And it turns out in this molecule, what we have is a carboxylic acid positioned so that the hydrogen on the carboxylic acid hydroxyl can fold around and be in a position to react directly with the nitrogen of the lactam. It does this actually through a cyclic transition state where when that bond is forming, it forms this ring. And what we see is that we can do intramolecular acid-base reaction with cyclic transition states when the transition state is a 5-membered ring or larger. If you look at the positioning of this carboxylate, it allows formation of a 5-atom transition state. So, in summary, what we have is both protonation to make this a better leaving group, plus the ring strain from the 4-membered ring to make this a better leaving group. So effectively, when that nucleophile pushes in there, it traps itself. You push in, tetrahedral intermediate pushes out, and it pops open that ring faster than the nucleophile can leave. And now that nucleophile has this group attached to it, and it's no longer able to function as it needs to function in the enzyme. Now, I'm not absolutely certain whether it's inactivating that nucleophile that causes the enzyme to be deactivated in this particular case. It could also be that by putting this large carbon group there, we block approach of the molecules that are needed to go together to do the enzyme reaction. But nevertheless, the effect is that we've neutralized this enzyme. So if we put a large amount of this in and neutralize all these enzymes, then the bacteria will not be able to build its cell wall, and therefore it will become vulnerable to our body's defense mechanisms, and our body can successfully fight off the infection.